Recently, we have discovered that the science of human aging is really pretty simple. We age because our cells divide and our telomeres get shorter. What's a telomere? Well, to answer that, let's zoom into a human's 100 trillion cells. Every cell contains a nucleus with genes and chromosomes. If you zoom in further, you see that the chromosomes are made up of DNA molecules that are 100 million bases long, coiled up like a slinky. There are long, repetitive sequences of DNA at the end of each of our chromosomes. These sequences are called telomeres. When a cell divides, the genetic material inside that cell needs to be copied. This is called DNA replication. During this process, enzymes that replicate a strand of DNA are unable to continue replicating all the way to the end, which causes the loss of some DNA. And the ends, if you remember, are where the telomeres are. At birth, we have about 10,000 bases. But as we age and our cells divide again and again, we lose those bases. And at 5,000 bases, we begin to die of old age. As telomeres get shorter, humans begin to experience the general effects of aging. Loss of muscle, failing memory, poor eyesight, lack of energy, and slower recovery after exercise. The bottom line is this. When cells divide, telomeres shorten. And bad things happen when telomeres get short. As an analogy, think of DNA as a long row of bricks, and of DNA replication as a bricklayer walking backwards on top of a brick wall, laying a new layer on top of that row. When the end of the wall is reached, the bricklayer finds himself standing on top of the bricks he's supposed to replicate. But since he can't put down a brick where his feet are, he steps back and falls off the wall, leaving the very end of the wall bare. As a result, the new copy of the wall is shorter. Just like this brick wall was copied imperfectly, our DNA is unable to copy itself perfectly. When a strand is replicated, the new strand is shorter than the old strand. Research we completed in 1997 established that humans also have an enzyme called telomerase that can lengthen telomeres. This research was so significant that in 2009 it was awarded the Nobel Prize in Physiology or Medicine. Let me explain this research further. We know that there must be a way for our bodies to re-lengthen telomeres because our reproductive cells do not exhibit telomere shortening and show no signs of aging. The reason these cells are essentially immortal is because reproductive cells produce an enzyme called telomerase. Telomerase acts like an assembly line inside our cells that adds nucleotides to the ends of our chromosomes, thus lengthening our telomeres. In a cell that expresses telomeres are lengthened as soon as they shorten. It's as though every time the telomere clock inside our cell ticks once, telomerase pushes the hands of the clock back one tick. Returning to the analogy of the bricklayer that can't lay the last brick on the brick wall, telomerase would be like an angel that flies in and puts the last brick in place. The telomerase gene exists in all our cells. That's because the DNA in every one of our cells is identical. A skin cell, muscle cell, and liver cell all contain exactly the same genetic information. So if the cells that create our sperm and egg cells contain the code for telomerase, every other cell must contain the code as well. This leading-edge science has created exciting possibilities in the field of anti-aging, possibilities that were only dreamed up until the impact on telomerase on aging was discovered. The reason most of our cells don't express telomerase is that the gene is repressed in them. However, if the appropriate compound, the right botanical, phytonutrient, or combination is found, science may be able to assist the body to de-repress telomerase, turning on the gene that's already present in every cell in the human body. I lead a team of scientists pursuing this line of research, and we are virtually the only scientists pursuing it. We have made significant progress in understanding the underlying science behind telomerase induction. We have studied and tested more than 300,000 drugs, chemicals, and compounds to document their effect on supporting telomere health. My biotech company developed the only high-throughput screening system in the world that is capable of screening for telomerase inducers. Our recent collaboration with John Anderson's 30 years of formulating experience And our strategic alliance with Isagenix's decade of proven transformational results is yielding results beyond anything we have ever experienced. It won't be surprising to see many companies trying to enter this space and begin marketing products touted to support telomeres. But I know from my research, not all telomere products are the same. One company is leading the way with a complete system that not only addresses the causes of aging, but also provides a credible product system to effectively support healthy telomeres. The first step to improve your chances of living longer and healthier is to control factors that are known to affect aging, such as toxicity, obesity, poor dietary and exercise habits, and oxidative stress. The next step is to assist the body with science-based, targeted nutrition to provide telomere support. I have dedicated my life to exploring the science of aging and how we might slow the aging process.